Hi everybody, Mrs. Singerly here, and today we're going to continue learning about carbohydrates. This is part of our biochemistry unit. Yesterday we talked about monosaccharides and disaccharides, and we even learned what a polysaccharide is. And so what we're going to do in today's lesson is we're going to talk about specific examples of polysaccharides, complex carbohydrates that you need to know for the AICE biology exam. And the first one we're going to talk about is starch. So starch is a molecule, a polysaccharide, that's made by plants. And the reason why plants make starch is they use it as a way to store all of those extra glucose molecules that are made during the process of photosynthesis. So remember that plants, they do photosynthesis, meaning that they're taking in sunlight, carbon dioxide, and water and using that to make food in the form of glucose. They also release oxygen as a byproduct of that process, but they're using it to make food in the form of glucose. The problem with having all of these random glucose molecules all around is it's not really an efficient way to store them. And by putting them together, bonding them together in a polysaccharide, is going to make it a compact way to store all of that extra food that you can use for later, because you have to use that food as an energy source to do all the things that plants need to do to keep themselves alive. Now, for us, for animals, and humans are animals, when we eat a starch molecule, so even things like, bread and pasta, all of those things ultimately come from plant products, right? And so when we eat those starchy molecules, we're going to break them down through digestion, which we call hydrolysis, which are going to take this long polysaccharide and we're going to break it back down into the individual glucose molecules, which we then can use for, say, like the process of respiration to make energy so we can do all the things that we need to do to keep us alive. Now, starch itself is actually a mixture of two substances called amylose and amylopectin. And they're made out of alpha glucose molecules. Remember alpha glucose from our lesson yesterday? Alpha glucose means that the hydroxyl is pointing which way? Is it pointing up or is it pointing down? Alpha is thumbs down. So alpha glucose molecules have the hydroxyl group below the plane of the ring. And so starch, like I said, is a mixture of amylose and amylopectin. Both amylose and amylopectin are made out of alpha glucose molecules. Now, sometimes you'll see it written that they're made out of what they call like these 1,4 linked alpha glucose molecules. If you see that, all that has to do with is the number of the carbons. So they number the carbons after the oxygen in this carbon ring. And so like the first carbon, we're going in a clockwise direction here. The first carbon is the number 1 carbon. Go down here, this is a number 2 carbon. This is a number 3 carbon, number 4 carbon, number 5 carbon. We're going to do this when we take a closer look at DNA as well. But that's all they mean by 1 to 4 linked, is where the glycosidic bond is occurring between which 2 carbons. Okay, so don't get too freaked out by that. We really want to focus on that. Starch is made out of amylose and amylopectin, and they're made out of alpha glucose molecules. Now, amylose is a straight chain. It's just glucose bonded to glucose, bonded to glucose, bonded to glucose, and it can kind of coil up a little bit, like what you're seeing in this picture here. Amylopectin is a branched chain of alpha glucose molecules. And so what you're seeing in this picture down here, right, they're kind of like a chain, and then there's this like branch that comes off of it, and that's what we mean by branch. Here you can see amylopectin as well. We kind of have this chain of glucose molecules, and then there's a branch coming off of it, and that's really what you need to know about these two things, is that they're made out of alpha glucose molecules. Amylose is a chain of glucose molecules and it can coil, but amylopectin is branched. And the reason why it's important that amylopectin is branched, and most starch is made out of mostly amylopectin, is that it provides more surface area for these molecules called enzymes to come and kind of break those down very, very quickly when the plant needs the energy to do its life functions, to keep itself alive and respond to its environment and grow and reproduce and do all of those kinds of things. And it can happen much faster if you have more surface area. Being highly branched also means that you can make it more compact, which makes it easier to store. Now, this mixture of amylose and amylopectin, it forms starch, and starch can actually be seen in grains. These starch grains are actually stored in organelles such as chloroplasts, and there's even these other ones called amyloplasts. And now we can kind of know why it gets its name as an amyloplast, because it's storing starch, which is made out of amylose and amylopectin. And so in a potato, which we call a tuber, or in what we call like the seeds of cereals, which would come from, a plant such as wheat, the starch is stored in grains there. And when we put it under the microscope, you can actually see it. So this is a compound microscope. This is the type of microscope that we would use in class. And these are kind of the starch grains that you can actually see under the microscope that you would find if you take a potato and you rub it on a glass slide. This is what it would look like under the microscope on high power, which is pretty neat. Now, the alpha glucose is also important for us humans, because they are easier to break apart. We said that they were highly branched, which makes it easy for plants to break them apart if they need the energy, but it also makes it really easy for animals to break off those glucose molecules and digest them to make energy. And so as humans and other animals, we have these enzymes, and I usually picture enzymes as little tiny Pac-Man that can go in, recognize their shape, and break it apart and digest the polysaccharide into those monosaccharides that we can then use as an immediate energy source. Now, glycogen is another polysaccharide that is used to store all these extra sugars. But glycogen is something that's made by animals. And that, again, includes humans. Humans are animals. And we make glycogen, the same reason why plants make starch, is as a way to store all those extra sugars. So when you eat a bagel, when you eat pasta, and you're not immediately burning up those glucose molecules and running 10 miles or something, your body's going to store it for later, because maybe, you're going to, you have the early lunch and you're not going to get home and eat dinner until 7 o'clock. There's going to be a time where maybe you're going to need to pull from this stored energy source and have that energy to get you through to dinner. So many animals will make this glycogen. It'll remove any extra glucose molecules from the bloodstream and store it for later. And the organ that we tend to store it in is the liver. We also store glycogen in our muscles as well. Now, when our blood sugar gets low, like if you have like 11 o'clock lunch and you're not eating dinner until 6 or 7, your liver will respond by breaking down that glycogen, right, our polysaccharide, back into those glucose molecules and release it into our bloodstream so our cells can use it for an energy source. And this process of you eat a lot of sugar, your body stores it, and then when you don't have enough, it releases it, is a way to maintain this constant level of sugar in our bloodstream. And that is a really good example of homeostasis, maintaining these constant internal conditions. And that's one of these things that we want to do, is we want to try to maintain our blood sugar level throughout the day. And for people who have diabetes, right, that is a disease, right? And if you fail to maintain homeostasis, it can result in disease or death. And so for people who have diabetes, their blood sugar level is not being controlled. It's going really high or it's going really low, and it's not staying at this constant level, which leads to disease and symptoms. And with diabetes, if it's not controlled, can lead to death. Now, glycogen, right, has a similar function to starch in that it's a way to store all these extra sugar molecules. And in fact, glycogen is very, very similar to amylopectin. It's made out of those alpha glucose molecules, and it's branched. So very, very similar to amylopectin. Its difference is that glycogen is much, much, much more heavily branched than amylopectin is starch. And so if you look at these two pictures here, right here, starch and plants, here's our branched amylopectin, and here's glycogen. And you can actually see how much more branched it is. Now, why that's important. The more branched it is, the more compact it can get. And for animals, right, we have to do something that plants don't have to do. We have to move around. So it's really important that our energy reserves are super nice and compact so we can tote them around with us wherever we're going, right? They need to be super duper compact. And the more compact it is, the more surface area there is. And so there's lots of places where our enzymes, our little Pac-Man, can grab on and break this down into the individual glucose molecules that can go into our bloodstream and can be used for energy for later. Now, what's interesting with glycogen is it's also going to form granules, kind of like how we saw with the starch grains. And these granules can be visible in the liver and the muscle cells where glycogen is stored, and these again can be seen under the microscope. Now, what's interesting with some microscopic images is they'll add dyes, or what we call stains, and it allows us to see some of the structures better. So all of the really dark green spots are these kind of glycogen deposits or glycogen granules. Next polysaccharide that we're going to talk about is cellulose. Cellulose is made by plants, and it is the major component of a plant's cell walls. It is one of the most abundant organic molecules on the planet due to its presence in cell walls, right? So plants and things like algae are going to have cell walls made out of cellulose, and they have a very slow rate of breakdown in nature. The other thing that's good to know about cellulose is that it's permeable. So even though it's forming a cell wall, think like the walls of your house, they still allow water in. So if you're a plant cell, having a wall, it's great because it's really strong, rigid supports, right? So if you're a plant, it's what allows you to stand up nice and tall. But it can also allow water and things into the cell that you might need. So just in case, it's a little kind of review from middle school, this is a plant cell, and plant cells have this really strong cell wall that goes around the outside. And so plant cells and organisms like algae are going to have their cell wall built out of this cellulose. This is a microscopic image of an aquatic plant known as Allodea. And these green little blobs that you see floating around, moving around inside the cell, those are chloroplasts. Those are the photosynthesis, sugar-making parts of a plant cell. But then this white line that you see kind of going around here, that's the cell wall, kind of almost forming these like tiny cells or like these tiny little rooms. And this is an onion cell, like the onion that you eat, that you chop up and put in your dish. When you're eating food, you're eating cells. And so if you put an onion cell under a microscope, this is what it would look like. And you can see this kind of, these white kind of lines. Those are those cell walls. So why is cellulose such a strong, rigid molecule? It's a polysaccharide. It's also going to be made out of glucose. So if it's made out of glucose, why doesn't it act like starch or like glycogen? Because it's made out of beta glucose and not alpha, right? So remember, beta glucose, where is that hydroxyl group? Is it thumbs up or thumbs down? Remember, it's going to be the opposite of what you think. So beta is going to be up. The hydroxyl group is above the plane of the ring. So why is that important? If you put two beta groups next to each other, the hydroxyl groups no longer line up next to each other. And so, because they're facing up. And so what happens is, in order to get them next to each other so that way we can form this glycosidic bond, every other glucose molecule has to be inverted, has to be rotated 180 degrees. It's upside down. So if you take a look here, and when you're looking at this, look for the oxygen. So notice in this glucose molecule, the oxygen is facing up, and in this glucose molecule, the oxygen is facing down. So in order to get these two beta glucose molecules to link together, we had to flip the second one upside down. We had to invert it. And you can really see it in this diagram down here, where you can see, like, here's the O on top, O upside down, O on top, O upside down. So every other one is going to be flipped, and it just has to do that in order to get that glycosidic bond. So this beta, we have these beta glucose chains. Every other one is flipped upside down, and we're linking them together. Then, so here we've kind of got one kind of linear chain, we have another linear chain, we have another linear chain, and another linear chain of cellulose. And so we have these polysaccharides, these really long chains, but then what's going to happen, we have a bond coming back, a hydrogen bond can happen. And they can form hydrogen bonds with each other, forming these kind of cross links. And when they form all these cross links, they call them microfibrils. And then the microfibrils can form cross links, and we can get this really super strong cellulose molecule. And so here is a microscopic image of cellulose of a cell wall. You can actually see these strands of this really strong cellulose molecule, or what we call cellulose fiber. And these fibers are actually made out of these microfibrils, which are these cellulose polysaccharides, which are all tightly cross-linked together. And that's really what gives it its strength. And how strong is it? I'm sure you guys are all wondering this at home. It is almost equal to steel. The cellulose has a very high, what we call tensile strength. Tensile meaning that it's very, very difficult to stretch or break, can withstand really large pressures. So if you think of something like a steel bridge and how strong a steel bridge is, that is almost equivalent to the cell walls of algae, right? And if you think of, like, what is paper towel out of? Paper towel comes from plants. And what is it actually made out of? It's made out of cellulose, and that's what gives things like paper and paper towels its strength, is it's made out of these cellulose fibers. It's really neat. Now, the problem with cellulose is that because it's so strong, it can be very difficult to digest. And some of that has to do with that we don't have the right kind of enzymes, the right kind of Pac-Men to break down beta glucoses. Because remember, those beta glucoses, they're still going to have the glycosidic linkages, but because every other one is flipped upside down, gives it a weird shape, and we don't have the enzymes to get at it and break it down. And so when we eat plants, when we eat vegetables, and you should be eating lots of vegetables, because you get out of them, you get all of the nutrients, you get the vitamins, and you get the antioxidants, things like all the different pigments, like all the different colors are really important. Those are part of the antioxidants that you're getting that are important for your body to function correctly. But we can't digest the cellulose part, right? Because when we're eating veggies, you're eating cells. And so we're getting all of the other stuff, but the one thing we can't digest is the cell wall. And that's what's referred to as fiber. So when people talk about you need to eat more fiber, you need to eat more roughage, they're talking about vegetables, and particularly they're talking about the cellulose. And why that's important is, one, is it's going to help you go to the bathroom, right? So it's going to help you to defecate. If you don't know what that word is, you can Google it. And that is important for your colon health, and to keep your colon healthy, and as a preventative for things like colon cancer, which I know you're not worried about because you guys are young and you're in high school, but when you get old like me, those are things that you're going to think about. So you're going to want to be eating veggies for all the nutrients, but also to keep your gut health and your colon functioning the way that it should be. Now, why can, say, an organism like a cow eat all the grass all the grass they want. Part of that is they have multiple stomachs, and they have special microbes in their stomach to help kind of facilitate breaking down that cellulose. They actually have bacteria to help them break down that cellulose. Interesting, right? Now, the last polysaccharide, this is the last one, I promise, is chitin. And I don't know why, this is always kind of one of like my favorite ones. I'm not a huge insect fan, but this is what makes up the crunchy part of insects. Chitin molecules are going to be similar in structure to cellulose molecules, meaning that they're also going to be made out of beta glucose molecules, where the hydroxyl is above the plane of the ring. That means that every other one is going to be flipped upside down, and it means it's also going to form this rigid support structure. And what organisms use this rigid support structure to do is to make up the exoskeletons of arthropods. Exoskeletons are outside skeletons, excuse me. And so like we have humans, we have an endoskeleton, an inside skeleton. Insects, lobsters, crabs, they have exoskeletons, outside skeletons, and that's what that crunch is made out of. And again, I said earlier that I'm not a huge insect fan, but these guys, I don't know why, are my favorite. These are called we call them in science world, we call them isopods. You might know them as roly-polies or pill bugs, but they're real fun. If you find them, they like curl up into little balls in your hands, and it's really neat to see them do that. Now, chitin can also be found in the cell walls of fungi. So fungi are going to be things like portobello mushrooms, yeast. Those are going to be our fungi. They're decomposers, and they have a cell wall too, but their cell wall is made out of chitin, not made out of cellulose. But we still get that strong rigid support out of those beta glucose molecules. So this is kind of, I know it's a little redundant, this is exactly what I was talking about, that chitin's made out of the beta glucose molecules. It does have a nitrogen in there, but you do not need to know that for your AICE exam. And kind of the last thing I'll throw in here is that, fun fact, chitin is so strong that it can even be used as surgical thread, which is kind of neat. All right, and I think that is where we're going to end. That is the end of our notes on carbohydrates. So the next thing that you're going to do is you're going to go into Google Classroom, and you're going to find there's going to be a section called Test Yourself, and it's a Google form, and you're going to go through and you're going to test yourself on your knowledge of carbohydrates. So you'll go through and see how you do. We're going to talk tomorrow. We'll talk more about questions that you have, things that you need answered, and some more fun facts, okay? So have a great rest of your day. I hope you found this video helpful, and don't forget to send me questions if you have them.